What you see over here is what we had discussed in the previous lesson. That were the stages, those were the stages of C3 cycle. So in the topic of photosynthesis, we are dealing with the Calvin cycle, or otherwise known as C3 cycle. You know that radiocarbon dating was used in order to trace the path how glucose is being formed. Now recall the steps that we have studied. The first step was that there was this process of carboxylation that was taking place. So we have mention of carbon dioxide in this particular region of this representation that I have made. So that step would be the part of carboxylation. Second, we had talked that it involved glycolytic reversal. If you have seen the video quite recently, this will be a revision. If you haven't seen it recently, then this would be a reminder that you should go and see it again. It would be easier for you because the various reactions have been discussed in detail over there. Here there is only presentation, and we are going to see how this particular cycle is going to lead to formation of glucose. And the last one is regeneration of RUBP. Before I start with the RUBP, no, like this, phosphate, this is what we have. Now, before I start with the main cycle, you have to keep in mind that the actual reaction of photosynthesis had 6 carbon dioxide molecules. Over here in this entire representation, we are dealing with 1 molecule of carbon dioxide, and 6 such cycles would result in 1 molecule of glucose. This you have to keep in mind, and after understanding you can relate with it how it would happen. As I told you, because there are 3 stages, so first of all let's divide those stages. See, from here till here we have the stage of carboxylation. This entire series of reactions is nothing but carboxylation process. So we have carboxylation over here. See, as I told you, there are 3 steps. So we have 3 steps, but because they are represented in the form of a cycle, we need to distinguish what part of cycle represents which step. So this part from here till there, 1, 2, 3, 4. These 4 stages are part of carboxylation. Now the remaining one from here till here, we have glycolytic reversal. Now due to the shortage of space, I have not mentioned few stages over here, and I have left that part onto you because you know very well we have done glycolytic reversal, how it takes place beyond fructose 1,6-phosphate. You can easily remember what those remaining stages were. I should not write it over here because it could be confusing. You can confuse it with the enzyme that is involved over here. This is glycolytic reversal, this particular part. I'm writing it over here, if you can see it. As I have mentioned, this particular region is glycolytic reversal, while the remaining one from here, including this particular zone belongs to regeneration of RUBP. This region is regeneration of RUBP. So we get to regeneration. We get to know which stage is happening where. Now once we have understood that, we're going to move further. What is happening, how it is happening. 1 molecule of carbon dioxide in the presence of rubisco would combine with ribulose 1,5-bis-phosphate, which is a 5-carbon compound. See, before starting with it, again I would repeat, this is the same reaction, but it is represented in a cyclic form because there is a cycle which is taking place, that is a C3 cycle. As you can see, the first compound that is being formed after association with carbon dioxide is a 3-carbon compound, and that is why this is C3 cycle. We have discussed about that as well. Now what you have to see is how it happens in a cyclic manner. What are the steps which are taking place and at what stage they are taking place. Now you see carbon dioxide comes and combines with the 5-carbon compound known as ribulose 1,5-bisphosphate in the presence of enzyme Rubisco, which shows the carboxylase activity and leads to the steps which are involving the process of carboxylation. Now what are those steps? First of all, this ribulose bisphosphate would form two compounds, 3-phosphoglyceric acid. Because it is a 5-carbon compound, it combines with 1 CO2. It is bound to form a 6-carbon entity, but it breaks down and forms 3-carbon compound, 3-phosphoglyceric acid. And that 3-phosphoglyceric acid, do keep in mind, there would be another 3-phosphoglyceric acid which would be formed. So from here till here, you have to keep in mind that another parallel step is going on, okay? So we are going to keep that in mind. 3-phosphoglyceric acid, another 3-phosphoglyceric acid would be formed. We have not mentioned it over here, so I would write 2 because 2 of them would be formed. Now 2 of 3-phosphoglyceric acids, each one of the 2 would use 1 ATP and break down to ADP. In the presence of enzyme triose phosphate kinase, there would be formation of again 2,1,3-diphosphoglyceric acids, 2 molecules. Now these 2 compounds, they are going to get reduced because NADPH, the reducing agent, would come and form 1,3-diphosphoglyceric acid, combine with it, lead to the formation of glyceraldehyde 3-phosphate, which is again a 3-carbon compound, and do remember 2 of them would be formed because in the beginning it was 2 again. Now by the end of this stage, we have used 2 ATPs because there are 2 molecules. We have used 2 NADPH, remember it. Now this glyceraldehyde 3-phosphate, because there are 2 molecules, they would undergo different reactions. One of it would go and would react with aldolase and form fructose 1,6-diphosphate. Now you can say that this was a 3-carbon compound, this is forming a 6-carbon compound. How is it possible? So do remember that we are not starting from the original point. That means there is nothing in the beginning, 0. We don't start from 0 in any biological reaction. What we have over here is that this glyceraldehyde 3-phosphate in the presence of this aldolase takes 2 paths. Okay, what is the first path? That is dihydroxyacetone 3-phosphate, DHAP would be formed. This would get isomerized to this particular form, and in the presence of aldolase, this glyceraldehyde 3-phosphate would form fructose 1,6-diphosphate. Now this fructose 1,6-diphosphate would form fructose 6-phosphate, then it would form glucose 6-phosphate, and then later on it would be glucose. So we have glucose in this particular reaction by the end of it. How do we regenerate the RUBP that would again combine, be ready for combination with carbon dioxide? This entire step involves the regeneration of RUBP. Now what you have to keep in track, this red arrow, this glyceraldehyde 3-phosphate comes and combines to form xylulose 5-phosphate. Now where does it, from 3 it becomes a 5-carbon compound. How does it become a 5-carbon compound? Because there are steps involved inside. What are those steps? Dihydroxyacetone 3-phosphate, it gets converted to pseudoheptulose 1,7-diphosphate, then it gets converted to pseudoheptulose 1,7-diphosphate, it gets rid of its 1-phosphate by the activity of phosphatase enzyme, and then it comes and joins with the glyceraldehyde 3-phosphate. 2 of its carbons come and join over here. It forms the xylulose 5-phosphate. In the presence of epimerase, there is formation of ribose 5-phosphate because pseudoheptulose would lose 2 of its carbons to form xylulose 5-phosphate. This xylulose 5-phosphate in the presence of epimerase and the ribose 5-phosphate in the presence of isomerase would lead to formation of ribulose 5-phosphate. Then ribulose 5-phosphate needs one more phosphorus one more phosphate in order to form the same compound that was in the beginning, and over here we have usage of another ATP that gets converted to ADP in the presence of enzyme phosphoribokinase. This is the entire step. Now, when I was of your age, what confused me more was, till here it was okay. Alright, there is formation of 2 glyceraldehyde 3 phosphates. There is formation of 2 compounds. It is having only 3 carbons. How is it possible that it is leading to the formation of 7 carbon compounds, 5 carbon compounds? We start with 3, we end up with 6, then we have a 7 carbon compound, then we have a 5 carbon compound. How all this is possible? So do keep in mind that the final product over here is mentioned, okay? Using the enzymes, the final product is mentioned over here, which involves not just 1 carbon dioxide molecule, it involves 6 carbon dioxide molecules. 6 such cycles would take place, and then we are going to represent the compounds over here. Now, when we have to show it cyclically, we use 1 carbon dioxide. It is better to represent all the reactions in 1 carbon dioxide's entities, carboxylation reaction, instead of doing it with 6. It would be really confusing. You can simply imagine that all these resultant things would occur if it is 6 carbon dioxide. Okay, so you can make it 6 carbon dioxide over here, 6 CO2. Automatically this becomes 12. This would also become 12. These would be 12. Now you can relate with it, that it is possible, because from there where there were 12, we have to come over here where there will be again 6. 6 and 6. Now probably you can relate with it. This is a little bit of mathematics and understanding that the carbon dioxide single is represented over here in the cycle, but in totality it is 6 carbon dioxides which come and undertake this reaction and leads to the formation of 1 glucose molecule, which I have skipped mentioning over here due to the shortage of space. So this fructose 6-phosphate would ultimately lead to the formation of glucose, which is a 6 carbon compound. We over here are representing the 6 carbon compounds in terms of 1 carbon dioxide. Please remember that. Now, by the end of this lesson, what is important, that how many ATPs and reducing powers have been used. This you have to keep in mind. Now see, over here we had 12 ATPs that were used, 12 NADPH which were used. Now when we come to this place, then instead of representing it 12, we have 6 ATPs which have been used. So in total there is usage of 18 ATPs and 12 NADPH for formation of 1 glucose molecule. Please remember this. So this is all you have to remember in the process of Kelvin cycle. You have to learn this process by heart. This is the exact representation as you find in your books. The enzymes are to be remembered by heart. This part, if it confuses you, I would suggest you to cram it, but do understand what is behind it. Again, I would repeat, just to end it up, that this glyceraldehyde 3-phosphate comes and goes through these 3 different passages. Okay, this I haven't mentioned over here. This comes and joins in fructose 6-phosphate as well. Okay, from everywhere there is addition of phosphates into this. So from everywhere when there is addition, you can primarily remember that this glyceraldehyde 3-phosphate is going to lose one of its carbons to fructose 6-phosphate one by one. It comes and drops one of its carbon over here, then it remains 2 carbon compound. The second glyceraldehyde 3-phosphate, which had converted into its isomeric form, it gives its 2 carbons over here through this passage. It is not possible for it to give it directly. Okay, we have to arrive from a 6 carbon stage which is scattered into 2 carbon compounds, 2 glyceraldehyde 3-phosphate compounds we are having. We have to make sure that one of the compounds drops its single carbon into glucose, because glucose would be formed when there would be 6 carbons. We had addition of 1 carbon. None of these is going to get rid of its carbon, because it is going to change it. So this carbon has to fall in here, over here at this stage. Supposedly you have put this carbon dioxide in a bucket and you move from one stage to another. You're changing the bucket every time, but it is carrying a carbon dioxide, and at this stage that carbon dioxide is to be dropped, which was picked at this stage. Now when it is to be dropped, it is to be dropped from this bucket, basically, this entity. Now these two compounds, glyceraldehyde 3-phosphates, two compounds, two of them, they come together and they are supposed to drop it over here, but it is not like this which is happening. You can't simply come and drop the carbon and ask it to form glucose. It takes two passages. First of all, it forms glucose 1,6-diphosphate. It goes with it. It takes this passage and forms xylulose 5-phosphate. Now, two of the glyceraldehyde 3-phosphates, one of the carbon is dropped over here. The remaining 5 carbons would combine to form this particular entity. And how it is formed? One of it takes this passage, one of it takes this passage. Alright, this is to be remembered. This is going all together differently. This glyceraldehyde 3-phosphate is not particularly converted into fructose. It takes this passage. Over here it drops its carbon. You have to remember this. This is going otherwise. This reaction is going otherwise. So that is why I have used the red arrow representation, which actually is the involvement of the compounds in the Kelvin cycle itself. This is going differently, but because at one stage you have this fructose 6-phosphate which is involved within this Kelvin cycle, so that is why we associate glycolytic reversal involving all these steps. This is all about Kelvin cycle. Have a look at it. This is what you have to remember. Why I had chosen to represent the reactions first and then do it graphically. The reason is that you have to remember the reaction, what converts to which compound, and once you have remembered that, it would be easy for you to understand that why these are being formed and what is the passage that is being taken by different compounds in presence of what all enzymes. So this is all about C3 cycle.